All right. Thanks, everybody. Welcome to the March ATP-Bio public webinar. Today's topic, let me get the slides running here, is going to be on CryoFoundry, which is an exciting initiative we have within the ATP-Bio ERC, supporting our startup culture. So again, you see here, for those who are not that familiar with ATP-Bio, we have represented here all the member institutions. And we have members from a couple of different institutions here today presenting on CryoFoundry, and you'll meet them today. So we have Linnea Warburton from UC Berkeley, and Luc Boudreau from MGH, and Carla Pavone, who works with me on the innovation ecosystem here on staff with ATP-Bio. So for those who are not that familiar with ATP-Bio, I'm going to just do a couple run-through slides on what it is. The vision of ATP-Bio is really stopping biological time, and it's really the impact of the technologies being developed by all of you here in the center, and as well as our member partners, really has the opportunity to advance in a big way and have impact in human health, biodiversity, food supply, and even more. So I think that's the important thing, is to think about this vision that the center wants to accomplish over its existence, and that impact is really through translation. And when we start to think about technologies and turning them into real life solutions, that translation occurs in partnership with innovation ecosystem, which is what we try to do and what we try to promote as a group. And if you think about vision, what are the things that we're trying to do? And these are slides that John Bischoff showed during our NSF site review last November, just to give you an example of the big things that this center can do. And it's important to think about this, that we have the opportunity to have impact in a big way across many different sectors. So if we think about the organs, organs waiting for patients, rather than the other way around, certainly is a paradigm shift in how we think about transplant. We think about cell therapies, regenerative medicine, the opportunity to use cryopreservation to pool and preserve things like islets, and be able to control type 1 diabetes without insulin injections. In the biodiversity area, you can see this great slides here from the idea of the lunar biorepository, again, an area in kind of biodiversity and preservation of species. This is a huge idea, right? And things that we can think about translating out of the center and having a major societal impact. So just to give you an idea that this is a center with big ideas, with big things, big impact, but a lot of it has to happen in how we actually take and make this impact happen. And our strategy behind this is really, again, with the goal of maximizing our positive societal impact, we do it through three things, right? Taking the technologies and making them ready for commercialization, connect commercialization to partners, to stakeholders, and then developing workforce capability for early phase commercialization. So that's kind of been our strategy, and over the last few years, and our idea behind the next few years is to really emphasize and execute on all these strategies. And among them, one of the components here is the startup culture under workforce. And really what we try to do in CryoFoundry is to help develop this idea of a startup culture within the center. So we invented this thing called CryoFoundry. It's actually coined by Carla Pavone. She actually has invented this terminology. I think it really captures a lot of this idea of taking cryobiology and cryopreservation, and this idea of foundry captures so many different images, but it's about this idea of forming ideas and making them into something. And so I thought I'd spend a little bit of time talking about what CryoFoundry is and what CryoFoundry is not. What CryoFoundry is, right? So it's a place for trainees and others, PIs, to present their technical ideas with an eye towards commercialization. It's an opportunity to find industrial member partner mentors. So we've had situations where we present in front of our industrial advisory board, and board members might say, "Hey, I'm interested in that. Use me as reference, or talk to me, call me up if you have questions about things." It's a venue to practice presenting, something that's not research focused. I think a lot of the trainees do a lot of presenting on their research, but this is an opportunity to think about things and present in a different way about their ideas. And it's also part of our quarterly industrial advisory board member partner meetings. So the thing what CryoFoundry is not, right? So it's not a pitch competition. Oftentimes there's a lot of opportunities where people pitch and they compete against each other for, prizes or dollars or what have you. This is not really meant to do that. It's more of a, I would say, a supportive environment to just try things out. Things don't have to be, fully baked, right? And there's no obligation to start up a company based out of anything you present here. It's not formalized. We don't have a set of slides you have to put together. There's no requirement for financial, deck that goes with your company idea. It's really about this formation idea. And it's also not a funding mechanism, although you never know. Maybe someone among the industrial advisory board or others might find an idea intriguing enough to think about putting investment in. And the last other thing that's really cool about is CryoFoundry. You get some really good swag. This is the lunch tote that Carla designed. We've been giving these out as swag during several of our meetings, and people really seem to enjoy those. Um, so with that, I'm going to introduce then Linnea. Linnea, I'll have you go and share your slides. Linnea comes to us as a postdoc out of Berkeley, and we'll talk about CryoVox. So Linnea, take it away.
Great.
Stop sharing.
Great. I'll share my screen.
Yep.
Hi, everyone. My name is Linnea Warburton, and today I'll be presenting about my work with CryoVox Technologies. At CryoVox Technologies, our mission is to reduce the drug failure rate by expanding access to 3D cell culture. Specifically, we have solved some problems in the supply chain in order to make 3D cell culture a shelf-stable product that can get to the customers that need it. But let's start with the problem. Drug development is incredibly expensive, and one of the reasons that it's so expensive is that it has a high failure rate. 90% of drugs fail during clinical trials. There's a big disconnect between preclinical drug discovery and clinical trials. And the reason for this disconnect is that the animal models that are used to test the drugs during preclinical drug discovery aren't predictive of human results. And one study found, they looked at 221 animal studies and found that they only predicted the human results 50% of the time. And so for many decades, the industry has wanted to find alternatives to animal testing, but there hasn't been a great alternative available. Recently, in the past decade, there's been a lot of really important progress with 3D cell culture with human cells. And when I talk about 3D cell culture, I'm referring to culturing cells in a 3D environment, such as in organoids or organ-on-a-chip systems, or in 3D bioprinted scaffolds. And these 3D cell cultures with human cells are far better at predicting human outcomes. One economic analysis found that if the pharmaceutical industry were to adopt the use of just 3D liver models, it could save $3 billion a year. And so 3D cell culture has huge potential within the industry, but most pharmaceutical companies haven't adopted 3D cell culture as an alternative to animal models. And the reason for this is that the current products on the market aren't meeting customer needs. And so that's where we come in, because we have technology that can turn 3D cell culture into a shelf-stable product that can be easily shipped to the customers that want to use it. Our patented manufacturing platform is for cryopreserving 3D cell culture. And once 3D cell culture is cryopreserved, it can be stored for short-term or long-term periods, and it can be shipped across the globe. What's crucial about our technology is that we see a 0% drop in cell viability as larger sizes are printed. Additionally, we've shown that we have the ability to cryopreserve stem cells and primary cells, which are the cells that are most sensitive to cryopreservation and also the most crucial to use in 3D cell culture. What is the value that our technology could bring to the market? Well, currently, if a pharmaceutical company wants to use 3D cell culture, they'll need to fabricate it in-house. This is going to require them hiring a trained fabrication team and putting a big upfront investment in terms of buying equipment, such as like 3D bioprinters or other organ-on-a-chip systems, and buying all the reagents for cell culture. And most importantly, this is going to take about 1 to 2 years for them to develop and validate this model. Instead, we say, outsource all of that work to us. When you buy a cryopreserved 3D model from CryoVox Technologies, we will already have developed and validated the model. So once it's shipped to you and arrives at your company, all you have to do is thaw it and it's ready to use. We have decided to start with the cryopreserved liver model, and this is for a couple of reasons. One is that unexpected liver toxicity is the #1 reason that drugs have to be withdrawn from the market when they're already on the shelves, and this results in a huge loss of revenue. Additionally, it all comes back to that 90% failure rate during drug discovery. 1 in every 4.5 drugs must be abandoned during clinical trials because of unexpected liver toxicity. And the economic analysis that I mentioned earlier found that adopting a 3D liver model would save the pharmaceutical industry $3 billion a year. What would our competitive advantage be in terms of 3D liver models that are already on the market? So as I mentioned previously, pharmaceutical companies have the option to fabricate these models in-house, but that requires a big upfront investment, and it also requires 1 to 3 years of developmental time. So instead, if they want to outsource this to a third-party company, one of their options is to buy the Emulate liver on a chip. This is an organ on a chip kit, and when it ships to the customer, they then need to assemble it themselves. This comes with challenges because it can be very challenging for the customers and the end users to assemble the organ on a chip system, and it can also be difficult with repeatability between users. So one of the only other options on the market is to buy liver spheroids from Inspiro. So this is a product, these are ready-to-use liver spheroids in a well plate, and they ship at room temperature. Now, one of the difficult things about hepatocytes is that they have a limited window of use, which is often 1 to 2 weeks. And so when you ship these liver spheroids at room temperature, one of those weeks is going to be taken up by shipping. Another problem with this business model is that the customers, when they order the liver spheroids, they then manufacture them on site. And so there's a couple weeks of lead time for the customers after they submit the order. Additionally, once the customers get the liver spheroids, they need to start their experiments immediately because that window of use is rapidly closing. So some of the solutions that we could offer with our cryopreserved liver models is that, like I mentioned, they're essentially shelf stable. They can be stored for short periods of time, long periods of time, and they can easily be shipped cryopreserved. And so as a result, it's possible to manufacture the liver model before the customer orders. So that way, when the customer orders, you can ship them out immediately and don't have to have several weeks of lead time. The window of use is not reduced by shipping, and the customer also has the option to store the models and not start their experiments immediately once they arrive. We have done over 30 customer discovery interviews at this point, and what we found is that customers are eager for this sort of solution. One person that we talked to was the director of toxicology at Genentech, and he said, when safety studies in primates miss toxicity, it wastes billions of dollars in 1 to 2 years for us. We're very eager to replace this step with in vitro models. And many of the people that we talked to were very eager for a cryopreserved model that would be convenient and save them time. Another customer said, if we could buy these models cryopreserved, it would be a huge time saver. If they use patient-derived cells, that would be extremely valuable. And ultimately, customers don't feel as if all of their needs are being met by the products on the market. One customer said to us, we tried using the Emulate chips for a while, but they were too complex to set up. We want something that's in a 96 well plate. Our plan is to start with a 3D liver market, a 3D liver model, and have that be our beachhead market. The specific application would be for pharmaceutical companies doing preclinical drug discovery. In addition, we would start with a liver model that is research grade, and then later on, perhaps would move to diagnostics grade or clinical grade, which would require a much longer approval process from the FDA and much stricter regulations. After establishing ourselves in the liver model market, we would want to expand into other markets, such as modeling other organs for preclinical drug discovery, such as the heart, the brain, lung, kidney, etc. We would also want to expand into other markets such as toxicity testing for other products or cosmetics testing. Now, our technology is doing something that previously was not possible. And so there's also the possibility of expanding into markets that are currently very small or don't yet exist. And so one possible market that we want to explore is cryopreserved tissue for emergency tissue transplantation. In addition, we would want to explore the expanding market for in vitro models for patient-specific medicine. We have been developing this technology within academia as part of ATP-Bio for the past 5 years. And as part of ATP-Bio, they helped us recognize the fact that this technology has commercialization potential. And so one of the things that we did this fall is we participated in the NIH Entrepreneurship Bootcamp. During this program is when we conducted our over 30 customer discovery interviews. It was also during this program that we identified that the liver model is a potential beachhead market for us. Since then, we worked with the Tech Transfer Office in order to get an option agreement for our issued patent. More recently, we've been participating in the Startup Legal Garage. This is a program through UC Berkeley and the UC Law School, and we've been working with law students and a patent lawyer to do a freedom to operate analysis, as well as a patentability analysis for other provisional patents that we want to submit. Also, more recently, with the help of Carla Pavone and Dan Chen, we applied and were accepted for the NSF I-Core program. In terms of the future for CryoVax technologies, we have what would be some short-term goals and some long-term goals. In the short term, our main focus would be developing the 3D liver model and optimizing the manufacturing process. We would want to collaborate with the Berkeley Drug Discovery Center so that we could start assessing the efficacy of our 3D liver model by screening it with various drugs. We also would want to focus on submitting a phase 1 SBIR grant so we could start moving out of academia towards doing research in industry. Within the first year, we would want to form corporate partnerships or first customers so that we can start demonstrating to the industry the advantages of using our technology. In terms of long-term goals, one of our most important challenges is identifying a cost-effective cell supplier. And so that's something that we've been thinking about very carefully. One thing that we have thought about is potentially partnering with the organ procurement network so that we could potentially use patient-derived cells from organs that were deemed not suitable for transplantation, but may still be useful for our purposes. Additionally, long-term, it would be ideal to try to partner with a pharmaceutical company that is doing testing to try to get their drug greenlit for a clinical trial. This would, again, help us demonstrate to the industry that efficacy and the advantages of using our product. And then in terms of long-term exit strategy, as for many life science companies, potentially we would want to look into acquisition by a larger company. One example of a company that acquires a lot of similar life science companies is the BICO Group. CryoVox is founded by myself as well as my PhD advisor, Dr. Boris Rubinsky. We've been working on this technology for the past 5 years at UC Berkeley, and we've been supported by both in funding and community by ATP-Bio. All right, thank you everyone for listening.
Great, thanks, Linnea. Excellent presentation. It's been so fun to watch the development, especially kind of your storytelling behind CryoVox develop over the last just the last 5-6 months. It's just every time you get in front of people and talk about it, just gets sharper and sharper. I just had one quick question, which was, in patient-derived cells, especially, we're taking them from patients that might have, compromised organs and so forth. How do we think about kind of standardization, or do we worry a lot about variability in those cells from a quantitative analytical point of view?
Yeah, thank you so much, Dan. And thank you both Dan and Carla, who have been so instrumental in sharpening the presentation as the months have gone by. So for your question, I think that is a really, really big decision to make, because on one hand, we could use iPSC derived hepatocytes. And with that, we would have much more reliability, and we could expect very similar results each time we made the model. But customers have expressed a lot that they really want they want primary human hepatocytes. They want patient-derived cells. And so there's so much variability between patient-derived cells. And so one of the possible approaches is that like many companies that are selling like in primary human hepatocytes in suspension, a lot of times they'll sell like pooled donor. And so my understanding is that the more donors you have, the less variability you'd expect. So I've seen products on the market like 10 pooled donors, 100 pooled donors. So I think if we were to go in that direction, such as like with the organ procurement network, it would be important for us to try to pool as many donors as possible to try to reduce variability in that way.
That's terrific. Yeah, no, that's a great idea. The idea of pooling certainly, kind of averaging out the variabilities is a great idea. So, for those online, certainly if you want to put a question in the chat, feel free to do so. We might have some time at the end, so we can pool questions. Back to pooling, pool questions for all our speakers. Or you can, if you want to raise your hand, we would be able to take a question or two right now, if anyone has one. Otherwise, thank you again, Linnea, for a great presentation. With that, we will introduce then Luke. Luke comes to us as a trainee from MGH, and he's going to talk about Cool Blood, which is a really exciting idea that they presented a few months ago at one of our IAB meetings, and now I'm back for more in an update. So, Luke, take her away.
Yeah, I always love going after Linnea because I call it a thought experiment venture because we have a lot of great ideas, but we're not quite in the space where we're going. Customers, customers, customers. But yeah, either way, it looks pretty though, which is nice. So yeah, I am here to present this venture that my boss Burke and I have been kind of working with Carla a lot. Called Cool Blood. But before we get into the nitty-gritty of it, I feel like I do have to go a little bit into the reasons why we want to even form a company called Cool Blood. So, let's see if my pointer works for me. And there we go. So first, before we talk about blood banking, we have to talk about Charles Drew. Charles Drew is the father of blood banking. Back in the 60s, when World War II needed more blood, he was the guy who was put up to the task of making blood banks and now allow us every day to have red blood cell transfusions, which have become the most common cell therapy and are often used just to increase oxygen concentration in the body to help the body keep using it. You need oxygen to make energy. About 120 million units are donated every year, but the need is constantly fluctuating, and the global demand is just rarely fulfilled for various reasons. And overall, we're seeing I believe it's a $40 billion industry. There's a lot of different figures out there depending on what you look at, but 40 billion is a nice number. Um, and the main problems we're seeing is that current storage parameters require cold stored red blood cells to be stored for only a maximum of 42 days after donation. So you can think that there's going to be a lot of waste. And only this one parameter known as hemolysis, or whether or not a red blood cell has lysed, is used as a quality parameter. So first, before I get into the nitty-gritty of my research, let's talk about a little bit of the modern approaches. There's currently two modern approaches. One, which I mentioned before, is cold storage, which allows you get high yield. There's that limited storage time, but it's easy to use because you can go straight from a freezer or really a refrigerator right into a patient. Alternatively, the other main use is freezing blood, which is really low yield, but you have a lot longer storage time. You're mixing blood with glycerols and a lot of other solutions to make sure it stays long and can withstand freezing. But it needs a lot of processing before use, and this was actually seen at 9/11 where they had a lot of blood, but not a lot of usable blood. So what we're trying to do is create a solution where we can store blood for longer periods of time, greater than 42 days, hopefully, without decreasing yield and quality from as we see at like the point of donation, such that it can be used for practical use like you would in cold storage. So our quote unquote product, or at least what we started with, was this idea of a two-pronged method of both novel additive solutions, or what we mix blood with to help it be stored in the short term, as well as supercooling, or the storage of blood below the regular freezing point, but without freezing. And we're using our base additive solution known as ESOL 5, which is alkaline and buffered, as well as chloride free, which overall just helps the samples stay better over a long period of time. And as you can see on the right, ESOL 5 was optimized both for cold storage and supercooling. But the difference with supercooling is we're seeing this ice formation. Overall, right now, we don't necessarily have a better supercooled solution with just hemolysis. As you can see, at the beginning, there's a lot more injury, but it levels out over time. So what we're trying to do is pull down that blue curve, which is supercooling storage, such that we can get prolonged storage with minimum hemolysis. However, we've done omics analysis and seen different effects with supercooled storage, and we're comparing a very bad supercooled versus a very good supercooled here. Overall, we see conservation of things like energy charge and 2,3-biphosphoglycerate, which allow for better release of oxygen and transport of oxygen into your blood system. So we're positing that these biomarkers, along with several other biomarkers, are showing that although hemolysis might not be as better at -4, there are certainly other parameters within these blood cells that will be helpful. Additionally, all these previous experiments were done at 1 mil. And so what we've done in the last few months have been scaling up our process. So rather than storing at 1 mil samples with a layer of oil on top, we've been able to store for up to 42 days in both that ESOL base solution, as we see on this left of the graph, and our proprietary additive solution on the right. We're bringing down that hemolysis. We've seen no ice formation, and overall, we're just having really good results. But, let's see. They won't let me go one more. Come on. But you got to ask, Luke, what does your company want to do? Because this is a really great product, but no one's going to give you the entire $40 billion blood market just because you have a reasonable idea. So what is cool blood? So over the course of the last few years, like I said, we've been doing all this research into supercooled storage. So we're combining several different lab IPs, such as the surface sealed storage technology that has allowed us to do storage at 1 mil over 42 at various temperatures, as well as these proprietary storage solutions that we're awaiting a provisional patent on to try to accomplish 3 objectives that I'll talk a little bit more about. The first of which is we want to create and produce these additive solutions in kits that you would see just a regular blood company might sell you to store blood. Second of all, we want to consult for the implementation of supercooled storage. We have great additive solutions that work at both +4 and -4C, and we're not going to be able to just make everyone move their freezers down to -4C. So in the process of this, we're also kind of trying to figure out consulting. And our third possibility is in building our own market. We want to be a part of contract research. Most of our resources right now are built for academics. So if we can, target smaller ventures and help them build, we'll be able to make our own market. And more on this in a second, because where do we fit in, right? This picture with me, this large red dot is the blood market. And as I said, it's big, but if you talk to Uzair, every time I say this, the blood market is very conservative, and for good reason, because it's blood. You just got to put in a person. Every time I say this, Uzair Raput from Instant Systems nods his head emphatically when I say it. And so it's built by, there's a very few, but very large companies that are in this field. So where does Cool Blood fit in? Currently in the regular market, I see a very thin slit that I'll tell you about in a second, as well as this almost building market that I'll tell you about as well. And that leads me to this first beachhead market that we've talked about a lot, and that's pediatrics, actually, because pediatrics relies on shorter storage because smaller people have a smaller surface to area volume ratio. things like calcium, lactate, and potassium build up at a smaller amount, but they can't take it as well. So this is our the market that's already there. Um, so what do we have at Cool Blood that could possibly be used, and we would like to explore with some customers very in the near future? We have lower lactate, slowed RBC metabolism in that first few months, and therefore we have a possibility of increasing that shelf life of regular pediatric transfusion blood from maybe 14 days to 21 days, which would be incredible for them. Additionally, our second beachhead is that one we'd probably have to help build, and that's with eRBCs. Now, what are eRBCs, you might ask? They're engineered RBCs. So RBCs, at the base case, are delivery devices. Regularly, it's oxygen, but you can add things like surface modifications, genetics, or encapsulated drugs in order to create new products. And that's been done to a degree for cancer therapeutics and autoimmune diseases with companies like Faxium, Scarlet Therapeutics, and Rubius Therapeutics, a couple of which have not done very well. Specifically, Faxium and Rubius Therapeutics have kind of either shelved their eRBCs or just gone under in the case of Rubius Therapeutics. So we're trying to target a third or another alternative eRBC with sort of more of a known money base, and that's DARPA. It'll let me screen. And DARPA, as of late 2024, put out a call for something called RBC Factory, where I'm going to dumb it down and say they kind of want to make super soldiers. In that they want to engineer RBCs to give soldiers in, augmented places to kind of have extra nutrients in the blood or drugs or prophylaxis. So the goal here in this beachhead market for us is to kind of target new ventures coming out of eRBC research or even research to help store and enhance samples through novel additive solution generation, possibly, because that's where our expertise is in a lot, and a lot of our IP can help, as well as optimize their storage conditions, whether that be 4C, -4C, -8C, or maybe even 20 degrees C. Who's to say? And that leads me back to that big idea of kind of what is Cool Blood, is we want to create additive solutions, foster that into contract research that maybe allows us to add supercooling into the supply chain, and therefore kind of build our market greater and greater into a nice venture. Like I said, we're in a very weird stage where we got one foot in, we got a lot of great ideas, but we haven't really done a lot of customer discovery like Linnea has. We have a huge team that's worked on this. Sachan and our friends at Alberta, as well as Anschutz, have kind of developed a huge pathway that have allowed me to kind of get in here and really take it from the reins and build their great ideas and put it forward in CryoFoundry that we've talked about earlier, that even allowed me to think about this and create what we now know as, I guess, Cool Blood. So thank you, everyone. I know that was probably a lot of information and not a lot of time. I will now take questions. Please
That was fantastic.
ask away.
Fantastic, Luke. Fantastic. We just want to just say again, just the progress you've made over the last few months. I know
Oh,
Carla's had a hand in it too, but
and thank you,
it's really
Carla,
great.
again.
Yeah, I
That
know
was the one thank you I
Carla's
didn't have.
behind a lot of the things, the advancements everyone's making. But it's really, I think, was fantastic to see that these kind of high value opportunities being identified. We do have one question in the chat. Let me see if I can get to it
That is
here.
from Gamid
Yeah, how
Abachev.
stable? Yeah, exactly.
Yes.
How stable is the system at supercooling temperatures? Do you have issues with spontaneous ice formation and crystallization? No.
So at -4, not really. Like I said, we've been doing this in samples up to 50 mil, 50 mils of packed RBCs. And we've done about, I want to say 40 blood samples over the last few months from 7 mils to 15 mils. At -4, it's very, very safe. We've had 0 nucleation events at that temperature over the course of the experiment. At +4, obviously, there's no nucleation. At -4, you definitely get into issues with nucleation, but I do this a lot in 1 mil samples. That's where we do the basis of a lot of our research, just to test additive solutions before we scale up into these larger systems. And over the course of my time doing this research, I don't think I've seen any at -4, and I've done hundreds of samples. If you add on that oil layer, which we have IP for, which we do our negative, there's another, I could talk about this for hours, but like our fast, rapid testing, we do at -8 with a layer of oil, and that generally like there's no ice formation under the oil, but at -4, definitely no oil. Granted, like I said, there's a level of like we would love to be able to have a hand in integrating supercooling because we do keep these stable on a shelf. There's no like crazy rocking or adding of energy into these systems that maybe even though they're in a sort of metastable state, though they're stable, could cause any ice formation. But yes, to answer the question straightforwardly, we have not seen any issues with crystallization at -4 for sure. Yeah,
Fantastic. We have one more other question which we'll take, which is from Laksha.
sure.
Hey, Luke, nice work. Does supercool system stability depend on scale of sample, meaning larger volume has higher chances of heterogeneous ice nucleation?
Like I said, we haven't seen any ice formation thus far. I think honestly, there might be less ice formation with the larger samples. There's a lot of like, academic research questions that we still have to ask and answer about those larger samples because, like I showed earlier in the uh, presentation very quickly, we've actually seen less hemolysis as you grow in size. So there's a question of do we get like micronucleation in the 1 mil samples, things like that, and that's far outside of the scope of our the venture. But no, Laksha, I don't think we've really seen any crazy ice formation.
Great,
Or yeah, sorry if that
great.
I didn't answer that fully, but yeah,
No, totally makes sense. Great. Thanks, Luke. And super great idea, interesting ideas. I like the DARPA idea of kind of like, the almost like blood doping, but for our super soldiers that'll be out in the field. It's a really interesting concept and among those high value opportunities that I think are gonna be the focus for you going forward. So thanks for that
yeah.
presentation.
Thank you
All
guys.
right. Very good. With that, we'll turn it over now to Carla Pavone. Again, works with me in the innovation ecosystem. She's an expert, and really an expert on entrepreneurship. Has mentored and helped. I don't know if it's probably hundreds, maybe thousands of people on their journey in entrepreneurship, and through her work here in the MBA school at the University of Minnesota, and elsewhere, certainly in I-Corps as well. So I just invited Carla to share some thoughts about, especially for a lot of the trainees who are looking at this combination of academic and entrepreneurial career paths, and this kind of combination for entrepreneurship career paths for researchers. So Carla, take it away. Can you hear you yet, Carla?
Carla, you're on mute.
Thanks, Bet.
See, I'm trying so hard to not make noise when I'm not supposed to talk. So thank you for reminding me. As Dan mentioned, I have worked with, in one way or another, literally thousands of generally early career researchers, meaning PhD students and postdocs and assistant professors, lab managers, et cetera. And for many of them, they go off and get jobs or they move into academia. Others do go into the entrepreneurship career path. And I've had about 45 different startups that I've worked with over the last 10 years or so. What I want to start with is this idea that there are a lot of different ways to get involved with startups. And part of what you need to think about is who do you want to be? Do you want to be like Christoph Krumm on the far right, who, like Linnea, is a CEO coming, and that point he came out of a PhD program in 2016 and is the CEO of a company that has raised about $7 million and is currently working not in the cryopreservation space, but in the renewable surfactant space, working with detergent manufacturers and other applications. And he's got a business with employees. He's got a scientific advisory board. He has business advisors. He has had no formal business training. And but he was very good at finding the resources he needed when he needed them, and then listening. And that was his goal. He wanted to be a CEO. Meanwhile, Andrew Allegria, I'm going to talk with you about in a little bit. He's a product guy. He's a science guy. He's an engineer. And he is vice president of robotics for Objective Biotechnology, and you'll hear more about them in a second. I could have mentioned several different professors who play the chief science officer role and who continue as professors. And many of you probably know who Alison Hubble is, and I'll talk more about her business. And then Abde Abnauer, who has a couple of different companies. But meanwhile, many of you have the opportunity to either work on technologies that end up being licensed to existing companies, and this is a physician in the Masonic Cancer Clinic here at the University of Minnesota, or at the very least, contribute to a technology and hopefully also get the opportunity to be a co-inventor, to have your name on a patent, because that really is helpful from a career perspective as well. So I'm going to share with you some examples. I mentioned Alison Hubble. Her company used to be called Blue Cube Bio. It's now called ATP-Bio. The person I want to highlight here is Rachel Leon. Rachel was a master's degree student in Alison's lab. Rachel was the first employee of ATP-Bio. ATP-Bio makes revenue now as a custom developer of cryopreservative solutions and consulting in this space as well, using technology developed out of Alison Hubble's lab. Over time, they've attracted other expertise. Most recently, a CEO, Jeffrey Allen, who comes out of kind of science-driven businesses, finance, and Greg Warshawski, who is basically the sales guy for this organization. But Rachel didn't have to know this all. Rachel is in her space, which relates to being able to actually deliver services and products to the customers of ATP-Bio. Objective Biotechnology is a recent spin out from the University of Minnesota as well. Andrew Allegria and Ame Joshi might be familiar names to some of the people in this group, because they've attended a lot of ATP-Bio events, and they were in labs that were part of both the Institute for Engineering Medicine and ATP-Bio. This is a robotics injection business. They have just closed on their first $1 million in investment. One thing to highlight is when you join a startup, you get a really fancy title because there's only a few people there. And they've between the $1 million of equity investment that they've recently raised, as well as SBIR grants, they are able to afford several of these people who are now full-time. Daryl, the chief science officer, still works at the University of Minnesota. James Grabeau, the CEO, deep experience as a salesperson in the research lab space, and Andrew and Emmy and Daniel really focusing in on developing and delivering product. Another thing I would highlight here is these guys started working with me in 2019. And Daryl and Andrew specifically. So this stuff can meander along for a long time. At that point, Andrew was a first-year PhD student. Within ATP-Bio, Industry Advisory Board, we have Revelia Bio, which came out of I think it was Princeton, no, Yale University, excuse me, a number of professors working in some way involved in this business. They are venture capital funded. They are the kinds of organizations that hire junior and recent grad and recent postdoc researchers into their operations. This is a startup that came out of a physician who was also a researcher at Mayo, who observed the issues that we're all very familiar with, the issues of cold chain. And his thing is around the container that biological materials are shipped with. And he started on this about 10 years ago, found himself somebody who could help him build it and develop the software that goes with it, not just the hardware. And over time, they've attracted other people as well. Steve Scully is, he's a CEO who knows a lot about his product, not a whole lot about his market, but he's managed to attract other people who can help him with that piece. And then finally, at the most recent annual meeting, you guys might have been exposed to Hunter from Cradle, which has a very ambitious goal, which is around basically, they put it pausing biological time for people, and so cryopreservation on a very large scale. And they have raised about $48 million in capital at this point, and are very much actively hiring right now. And these are the as of yesterday, the positions they're currently hiring for. So how do you think about this? The way you think about this is you want to build options for yourself. You want to make use of resources that are available to you, and you want to do it earlier rather than later. You guys are wonderful at doing your lab work and then getting so caught up in that, you don't necessarily think about the rest of it. So I'm going to encourage you to think about becoming a T-shaped professional with boundary crossing competencies. And if you want to read more, we have a PDF here about that. The idea of understanding how to communicate. And Linnea and Luke have come an amazing way since they first were communicating these concepts. The ability to develop strategic perspective, to develop networks, to do project management are just some of the examples. Learning the vocabulary of business would be another example. And then you're already developing depth and thinking about not just in terms of your discipline, but in terms of some ecosystem. So in our case, it might be around cryopreservation as an example, more broadly. Who are the players and how would you navigate within there? And how do you become a T-shaped person? A, you can talk with me, but B, there are all kinds of workshops and webinars you can be participating in. And this is one example. Linnea gave other examples out of Berkeley, and you can find, and in each of our institutions, there are other examples, but this one is one that I'm associated with that anybody from any university at the in the US could be participating in. Okay, so thinking about learning about other types of funding, learning about IP and working with a tech transfer office, learning about this I-Corps program that's been mentioning, developing partnerships with industry, understanding your range of career paths, which for many of you, your advisors may not be as familiar with because they are so immersed in the academic world. It really depends on the advisor. Okay. So your takeaways, think about who you want to be. Do this stuff earlier rather than later so you have a long enough runway and you have that opportunity to explore and maximize your options. Doing that T-shaped skill set and leveraging connections. Luke mentioned Uzair at Instant Systems. Dan can be talking with you about who else we have in the IAB. Linnea has done a bunch of outreach. I can be helping you guys as well. All right, thank you.
Great, terrific talk, Carla. And it is the numbers are amazing in terms of how many people and companies you've touched over the years. And just been really, really proud to see people advance in their careers and companies develop. I think a couple of things you bring up is, the T-shaped person is something that we look at not only for entrepreneurs but also large companies too. We've done a lot of talking about when I was at 3M, how do we want people to be in kind of agile teams, and that T-shaped skill set or personality is certainly very important. Another thing I wanted to reflect on is this idea of resources, and Pat's been chiming in here. Pat's with MNSBIR, who helps companies apply for and navigate the SBIR framework. A member partner, certainly there's companies or organizations like Pat's who can help trainees, PIs, others who are starting companies navigate the SBIR area. And there's, again, I think part of our goal as an innovation ecosystem is to provide member partners who can also step in and help. Pat, I see you came on camera. You want to chime in for a couple words on MNSBIR?
Well, of course, hello everyone. And Carla, always a pleasure to hear you speak. Always a wealth of information. So appreciate that. So MNSBIR stands for Minnesota SBIR. So we only work with Minnesota firms, but we are a partner with ATP-Bio because we want to support innovation and entrepreneurship across the United States. And if we can be helpful to any of the teams that are pursuing SBIR funding, we can at least provide some guidance and advice to you as you start down that path. And one thing that I've always said over the years is that after basic research, the next pot of money is SBIR and STTR, and it's all about translation and commercialization. And I'm happy to hear that, some of you already got that on your plan. So that's great. Just start early and be persistent.
Fantastic advice, Pat. Really appreciate that. Again, I think you'd be a great resource for a lot of people in the organization. So I appreciate that as well.
Yeah, well, one thing before I forget, there are we have sister organizations all across the country. So if you want to get connected to your SBIR resource in your state, just send me an email and I will get you in touch with the right group for your state.
Fantastic.
And I'll put my email in the chat.
Okay, fantastic. Great. Thanks, Pat. Great. Any other questions? Any questions for Carla that people have? All right. Otherwise, I will also, I see that Nicole put in a cheeky plug for the Society of Cryobiology. It's not that cheeky because it involves actually trainees from ATP-Bio. So I see we have Purva, Max, and Joe will be talking to that. So take a look at that in the chat, the webinar. It's a cross-promoted webinar with the Society of Cryobiology, certainly one of our important partners and also a resource for us as well. So I'll give everyone a last second to ask any questions or have any other interventions. All right. Otherwise, we can go ahead and just close for today. But thank you for joining. We'll try to do this on a periodic basis. It's just fun to see new ideas come up. And again, feel free to reach out to myself or Carla if you're interested about learning more about CryoFoundry, you want to present at the next quarterly IAB meeting, or just want to just chat and talk. So certainly Carla and I are available for that as well. All right. Thanks, everybody. We'll close it out there. Bye now.
Thanks, Dan.